Hello friends, in this video we are going to discuss about bacterial plasmids. Under bacterial plasmids we will discuss about water plasmids, then we will discuss about facts and terms related to plasmids, then properties and characteristics of plasmids, elements of plasmid as vector, role of plasmids, then types of plasmid on the basis of ability to transfer to other bacteria. Under this, we will discuss about two types of plasmids, conjugative plasmids and non-conjugative plasmids. Then we will discuss about types of plasmids based on function. Under this, we will discuss about 5 types of plasmids: fertility, F plasmid, resistance, R plasmid, Col Plasmid, Degradative plasmid and Virulence Plasmid. Then we will discuss about the term episome. So stay tuned. So first of all, what are plasmids? Plasmids are naturally occurring small circular extrachromosomal autonomously replicating fragments of DNA present in prokaryotic and few eukaryotic organisms. Extrachromosomal means these are present in addition to bacterial genome. Autonomously replicating, that is, these are self-replicating. These have their own origin of replication. Plasmid term was coined by Joshua Lederberg in 1952. These are not required for bacterium survival. That is, these are not vital for bacteria but carry genes that provide various advantages to plasmid host cell. It means that cell in which plasmids are present have advantage over cell in which plasmid is not present. Example, antibiotic resistance. If the plasmid present in the host cell has genes for antibiotic resistance, then this host cell is resistant to antibiotic. And the cell in which this plasmid is not present, these are not resistant to antibiotic, that is, they are killed in the presence of antibiotic. Now the facts and terms related to plasmids. First is size. Plasmids are much smaller than bacterial genome. These carry additional genes. These carry the genes in addition to the vital genes that are present in bacterial genome. These exhibit wide range of size varying from 1 to over 400 kilobase pair. Some are as large as 2 megabase pair. These are known as megaplasmids. Second is shape. Plasmids have 5 different types of shapes. First, nicked open circular. DNA has one strand cut. This type of the plasmid is double-stranded circular and its one strand has cut. Relaxed circular. DNA is fully intact. So it is double-stranded circular and it is fully intact. Linear DNA has free ends, supercoiled, completely intact but has a twist, hence smaller. This supercoiling we have already discussed in separate video. You can refer that video. The link is given in the description box. Supercoiled denatured. So these are also supercoiled but some part is lost due to denaturation. Hence, these are little smaller in size. Third is copy number. Copy number is the average number of molecules of a given plasmid per bacterial chromosome. And copy number ranges from 1 to 3 copies to 100 copies. Host range. Number of bacterial species in which typical plasmid can exist. Most plasmids have narrow host range. That is, these are present in limited number of species. Some have broad host range. It means these are present in large number of bacterial species. Fifth, compatibility of plasmids. That is, ability to coexist in the same bacterial cell. So if 2 or more plasmids exist in the same bacterial cell, then these are known as compatible to each other. But if they cannot coexist in the same bacterial cell, then these are known as incompatible. Now let us see Properties and characteristics of plasmids. First is extrachromosomal. So these are present in addition to the bacterial chromosome or bacterial genome. These are mostly double-stranded except linear ones. These replicate independently and hence these are known as replicon. Replicate independently means these are autonomously replicating or self-replicating within a suitable host. These are non-essential, that is, these are not vital for the survival of bacteria, but their presence is beneficial and may provide selection advantage, example as antibiotic resistance. So if there are certain bacterial cells which have the plasmid carrying the genes for antibiotic resistance and certain bacterial cells which do not have this plasmid. So in the absence of antibiotic, their survival will have no difference. But in the presence of antibiotic, the plasmid carrying cell has advantage over the cell in which plasmid is absent. That is, those having the plasmid which has genes for the antibiotic resistance can survive, but those in which the plasmids are absent, these are sensitive to antibiotics and hence these are killed. Plasmids are also known as sex factors, conjugants, extrachromosomal replicons or transfer factors. This we will understand better in horizontal gene transfer video. If we talk about size of the plasmid, their size varies. It has wide range of size which ranges from 1 to 400 kilobase pair and in some cases around 2 megabase pair. So this size varies from mini plasmids to megaplasmids. Copy number. The copy number range from 1 to several hundreds. Larger plasmids have fewer copy number, but smaller plasmids have higher copy number. These are generally circular, but linear plasmids exists and require specialized replication mechanisms. So mostly these are circular, but in some cases linear plasmids also exist and these require some special replication mechanism which is different from the circular plasmid replication mechanism. Those with low copy number have partition systems to distribute copies to daughter cell. Some can insert into host chromosome known as episome. This we will discuss in detail in next part of this video. There are certain plasmids which have no effect on phenotype of host cell. These are known as cryptic plasmid. Now let us see role of plasmids. Gene products encoded by plasmids are rarely essential for survival. So the gene products or the proteins which plasmids encode, these are non-essential for survival or these are not vital for survival but provide advantages to the organism such as utilization of unusual carbon sources. So these help in the utilization of certain unusual carbon sources like toluene, etc. Then resistance to heavy metals and antibiotics. Sometimes these provide resistance to heavy metals and antibiotics, and also synthesis of toxins and proteins that allow successful infection to higher organisms. Now the use of plasmids. Plasmids serves as useful genetic tools in molecular biology and genetics. These are used in gene cloning, recombinant protein manufacturing and gene therapy research. These can be utilized to introduce, manipulate or delete specific genes within a host cell. So these points we will discuss in detail in recombinant DNA technology video. Now let us see general structure or elements of plasmid as vector. Plasmid vectors helps in cloning small DNA fragments in bacterial cells. And the elements of plasmid as vector as shown in the diagram are origin of replication, antibiotic resistance gene, selectable marker, promoter, 5' primer site, restriction site, inserted gene, restriction site and 3' primer site. Ideal plasmid vector has these properties: small size, high copy number, origin of replication, restriction sites and selectable markers. In order to use natural plasmids as cloning vector, several modifications are required. Hence, what we can say that plasmids serve as essential genetic tool in molecular biology and genetic engineering. And these can be used to introduce, manipulate or delete specific genes within a host cell. These we will discuss in detail in recombinant DNA technology video. We are going to discuss about types of plasmids. So first of all, let us see the types of plasmids on the basis of their ability to transfer to other bacteria. On this basis, these are of two types. First is conjugative plasmids. These code for functions that promote transfer of the plasmid from one bacterium, that is the donor bacterium, to the other bacterium known as the recipient bacterium. So what we can say that these are capable of horizontal gene transmission. Thus, they perform conjugation. These have tragenes that help in the process of conjugation. Example, synthesis of sex pili, which is then later converted into conjugation tube and helps in the transfer of plasmids from donor to recipient. This type of plasmids are found abundant in naturally isolated bacteria. Large plasmids whose size is greater than 40 kilobase pair are often conjugative plasmids. Second is non-conjugative plasmid. These are incapable of initiating conjugation because they do not encode self-conjugative system. Hence, these may be transferred from donor to recipient bacterium in assistance with conjugative plasmids. Plasmids smaller than 7.5 kilobase pair are often non-conjugative plasmids. Now, types of plasmids based on function. On the basis of this, these are of 5 types. First is fertility, in short F plasmid. These are also known as fertility or sex factor. These determine the sexuality of bacterial cell. Bacterial cell in which F plasmid is present is known as F plus male or donor cell, and bacterial cell in which it is absent is known as F minus, that is female or recipient cell. These are transferred from donor to recipient through conjugation. Hence, what we can say that these are conjugative plasmid. These exist independently or integrate into main bacterial genome or chromosome. Therefore, F factor is an example of episome. A bacterial cell with an integrated F factor is known as HFR cell, that is high frequency recombinant. Functions of F plasmid carry genes, that is tragenes, to promote conjugation, that is transmission of F plasmid from donor to recipient cell. Second is resistance, or in short R plasmid. These confer resistance to one or several antibiotics. Some R plasmids have a single resistance gene. Then host cell is resistance to one antibiotics. And if R plasmids have multiple resistance gene, then host cell is resistance to several antibiotics. Hence, these are threat to medical sciences, because if the bacterial infection is caused by bacterial cell in which R plasmid are present, then these are resistance to antibiotics. Hence, this type of infection cannot be treated by that particular antibiotic. R plasmids are conjugative plasmid. It also provides immunity to harmful effects of heavy metals to host cells. Now, the third type of plasmid is Col Plasmids. Col Plasmids contain genes that produce bacteriocins. So now, what are bacteriocins? Bacteriocins are antibacterial polypeptides which kill different strains of bacteria. And how they kill bacteria? By inhibiting one or more essential process as DNA replication, transcription, translation, or energy metabolism. Bacteriocins that are produced by E. coli are known as colicins. Colicins inhibit the growth of susceptible bacteria, or in other words, sensitive bacteria. There are many types of colicins produced by different strains of E. coli, and these are designated by a letter. Example, colicin B, etc. And each inhibit the growth of bacteria by particular mode of inhibition. Fourth is degradative plasmids. Degradative plasmids helps the host bacterium to catabolize organic compounds that are not commonly found in nature, as xylene, toluene, camphor, etc. There are certain genes present in these plasmids that encode enzymes which degrade these compounds to produce carbon and energy sources. These are also conjugative plasmids. Degradative plasmids increases the chance of survival of host cell in the presence of these unique substances or in the presence of these unique conditions. Now fifth is virulence plasmids. Virulence plasmids can give pathogenicity to host bacteria, or in other words, turns that bacterium into a pathogen. What are pathogen? Pathogens are agent of disease or disease causing agents. Disease causing bacteria easily spread and replicate among affected individuals. Example, E. coli has several virulence plasmids. Similarly, Salmonella enterica also has virulence plasmids. While discussing F plasmid, we came across episome term. What are episome? Plasmids having the ability to integrate in the chromosomal DNA of host cell are called episome. So, plasmids which are capable of incorporating into the main bacterial genome or bacterial chromosome are known as episome. It remains integrated for prolonged periods and are replicated along with host chromosome during cell division. Eventually, it becomes essential part of host genome. Example, F factor. This F factor we have already discussed. A bacterial cell with an integrated F factor is referred to as an HFR cell. Full form, high frequency recombinant cell. This is because of high efficiency for chromosomal gene transfer. So, in HFR cell, chromosomal genes are transferred from donor to recipient with high efficiency. This we will discuss in detail in bacterial conjugation topic. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.